Hello and welcome back. In this video, we're going to continue into a new chapter, which is on Chapter 4, Biocatalysis. Once again, I'm Miss Delia, and let's go through the several subtopics that you will see in this chapter, starting with 4.1, which is the properties of enzymes and mechanism of action. So in this subtopic, we're going to learn about what are enzymes, what are they like, and how do they do what they're meant to do. Now, before we go into the lessons itself, here are the learning outcomes for this chapter. By the end of this lesson, I hope you would be able to state the properties of enzymes, state the 6 classes of enzymes according to the IUBMB classification, explain how enzyme lowers activation energy, and illustrate to explain the mechanism of enzyme action based on the induced fit model, and also state the factors that affect the enzymatic reactions. Now, if you're ready, let's get started with the first segment. We'll talk about the properties of enzymes. And of course, I will take this opportunity to first explain what an enzyme is. So if you notice, this chapter is called Biocatalysis. It's talking about a catalyst in nature, which is enzyme itself. An enzyme is a globular protein that function as organic or biological catalyst that speed up the rate of metabolic reaction by lowering the activation energy. So the keywords, if you need to give the definition of enzyme, is globular protein, speed up rate of reaction by lowering the activation energy. In case you forget, the activation energy can be defined as minimum amount of energy required by a reactant or substrate to start a chemical reaction. So here we have this very common graph that you would see not only in biology but also in chemistry, which is there are two curves here. The first curve, or the red one, is showing how to get reactants to become your end products, it needs to reach this activation energy. But if you have enzymes or catalysts present in your reaction, then it doesn't need to go all the way. It only needs to reach about this much of the activation energy. Now, when we talk about enzymes, they can be represented in any of the following forms. You can sometimes find books that show enzymes like this. So this yellow part over here could be how you draw an enzyme. You could also illustrate an enzyme like this. So in this molecular model of this enzyme, we have a 3D structure that shows every single element that is in this molecule. So setiap H, setiap carbon, setiap nitrogen, dan sebagainya boleh diwakilkan dengan biji-biji dalam gambar ini. We also have another form of illustration that we can use to represent enzymes, which is in this form. So a lot of higher levels of material, especially if you're talking about university level where they have conferences discussing this and that enzyme, this and that protein, they would represent it in this form most of the time. So this form is showing all the beta pleated sheets and alpha helixes that you would find on the protein structure. Now, all 3 of these diagrams are showing the same enzyme, just represented in different forms. All of these are of the enzyme catalase. Of course, this slide is not a key point. If people ask you to define enzymes, this slide is here so that if you ever see these things in any of the media that you consume after this, you will know that you are looking at a diagram of an enzyme. Okay, so let's learn more about the properties of enzymes. First things first, we can say that all enzymes are globular proteins and they act as a biological catalyst. They will be required in small amounts to catalyze the reactions. And the enzyme is not altered at the end of the reaction. That means they are reusable. So dia adalah protein. Fungsi dia adalah untuk mempercepatkan reaction. Diperlukan dalam kuantiti yang sedikit dan bentuk dia tidak akan terjejas atau berubah selepas reaction. So kita boleh guna dia semula. Enzymes are highly specific in action. And we would have one enzyme that will catalyze one reaction. Dia tidak akan tiba-tiba enzyme catalase ini buat function lain. Memang dia punya fungsi adalah untuk catalyze jenis reaction itu sahaja. Enzymes may catalyze reversible or irreversible reactions. So reversible reactions is things like, okay, after the substrate becomes the product, it may become the substrate again. Whereas irreversible reactions is once the substrate becomes the product, it can never go back to being the substrate again. The activity of enzyme will be at its maximum at its optimum pH and temperature. So setiap enzyme dia ada dia punya optimum pH dan optimum temperature. Maksudnya kalau dia sudah tercapai pH yang dia paling suka, rate of reaction paling tinggi. Kalau dia sampai suhu yang dia paling sesuai, rate of reaction dia paling tinggi. Enzymes will be denatured or destroyed at extreme pH and very high temperatures. So kalau suhu terlalu tinggi, enzyme denature, dia akan rosak. Kalau pH terlalu ekstrim, so sama ada terlalu alkali ataupun terlalu acidic, enzyme itu pun akan denature, dia akan rosak juga. So here we have something that illustrates how the rate of reactions of an enzyme would change with the temperature. So in this case, the optimum temperature is 37 degrees Celsius. Anything higher than 37 degrees Celsius, the enzyme will slowly start to denature. Anything lower than that, the enzyme will be fine, but the rate of reaction will be slower because there is less energy present. At low temperatures, the rate of reaction is slow due to the slow movement of molecules. And therefore, there is decreased chances for the substrate to collide with the active site of enzyme. Enzyme is also inactive at low temperatures. So, kalau ada orang tanya, apa terjadi pada enzyme pada suhu yang rendah, yang di bawah optimum temperature dia, kamu boleh explain dengan cara ini. Okay, so as the temperature increase, the movement of the molecules will increase, and you will have collision between substrate and active sites will also increase. And thus, the rate of reaction increase. For every 10 degrees Celsius increase in temperature, the reaction rate will be doubled, up to the optimum temperature at which reaction rate will be maximum. So, slide ini dia sedang explain apa terjadi bila kita tingkatkan suhu dalam reaction itu. So, bila suhu meningkat, that means the molecules will have more heat energy. Suhu lebih meningkat, molekul akan bergerak dengan lebih laju. Lebih laju means lebih banyak chance untuk substrate dengan active site of enzyme bertembung. So, rate of reaction increase. Dan setiap 10 degrees peningkatan suhu, rate of reaction akan meningkat 2 kali ganda. That is what the third sentence is saying here. Up until the optimum temperature. Sehinggalah dia tercapai optimum temperature untuk enzyme tersebut. Bila dia sampai pada optimum temperature, the rate of reaction will be maximum. And as I mentioned earlier, beyond the optimum temperature, the reaction rate will decrease due to the denaturation of protein. Limpas saja optimum temperature, lebih panas daripada optimum temperature, rate of reaction akan turun sebab protein akan denature. Dia start untuk rosak. This is because there is change in the conformation of active site. In case you haven't noticed it, whenever we see the word conformation, we are talking about change in shape ataupun referring to the shape of that molecule. So, when we say the protein denatures, bukannya bila saya cakap dia rosak, itu lebih kepada bahasa pasar. Apa yang actually berlaku kepada protein tersebut bila dia denature adalah bentuk molekul dia berubah sehingga dia tidak dapat laksanakan function dia yang asal. There is change in conformation that causes it to lose its function. The change in conformation of the active site of the enzyme is a result of the breaking of ionic bonds, hydrogen bonds, disulfide bridge, hydrophobic interactions, and van der Waals interactions. Kalau kamu ingat apa yang kita belajar pasal protein pada semester lepas, untuk protein itu sampai kepada tertiary and also quaternary structure, selain daripada peptide bond, dia juga akan ada hydrogen bonds, disulfide bridge, hydrophobic interactions, dan van der Waals interaction di dalam molekul dia sendiri. So, bila kita punya suhu terlalu tinggi, segala bond-bond selain daripada peptide bond ini akan terbuka, terurai. Itulah bentuk dia tidak betul sudah. So, because of this conformation, because of this breaking of bonds, the substrate can no longer fit into the active site of the enzymes, and so the enzyme will be useless. So this is once again showing the graphs, rate of reactions against temperature, and you can see where is the maximum rate of reaction right at the optimum temperature. Now for this graph on the right, you could see that there are two curves here. The black curve is showing the optimal temperature for the typical human enzyme. So for human enzyme, the optimal temperature is usually around 37 degrees Celsius. And over here, the red curve is showing the optimal temperature for the enzyme of thermophilic or heat-tolerant bacteria. And the optimal temperature for this is more or less around 75 degrees Celsius. Jadi apa yang saya mau tunjukkan di sini adalah optimal temperature bukan sama untuk setiap enzim. Biasanya dia berbeza mengikut spesies. So for humans, our body temperature is 37, so our optimum temperature is around 37. But if it's a different species, especially in a thermophilic bacteria, which usually lives in hot springs or near volcanic sites, then of course they can tolerate higher temperatures, and they will want to use enzymes that is suitable for the higher temperatures as well. So lain organism akan ada enzyme yang berlainan optimal temperature. So that's it that I want to talk about for temperature for now. We'll come back to it again later. Let's talk about another property of enzyme, which is about its pH range. So something else about enzymes is that enzymes function efficiently within a narrow pH range. Different enzymes will have different optimum pH. For example, we have the enzyme pepsin, which has optimum pH of 2. You can find pepsin in the human stomach. We also have the enzyme amylase, which has optimum pH of 7.2. You can find amylase in the salivary glands, in the liver, and in the small intestines. So both these enzymes are in humans. So it's in the same body, but they will have a different optimum pH because they are in different environments. The maximum reaction rate will be achieved at the optimum pH. A change in the pH above or below the optimal pH value will change the hydrogen ion concentration. So referring to this final point here, kalau pH kita rendah daripada optimum temperature, or as the pH is getting lower, it's becoming more acidic. That means you will have higher hydrogen ion concentration. If the pH is becoming higher, becoming more basic, lebih alkali, then that means you will have decreased hydrogen ion concentration. Why does the hydrogen ion concentration matter? It's because this ion will alter the ionic charge of the functional group of amino acid in the enzyme. Concentration of hydrogen ion akan kacau ionic charge. And thus, it could break the ionic and the hydrogen bonds that hold the specific shape of the enzyme. Remember what I mentioned earlier. For a protein to maintain its tertiary and quaternary structure, there are bonds other than peptide bonds such as ionic, hydrogen, disulfide, van der Waals, etc., other bonds that hold the protein molecule together. So when the pH value changes, concentration of hydrogen ion changes, ionic charge of the functional group will be altered, And thus, you could break the ionic and the hydrogen bonds. Asalkan bond ini terputus, bentuk kita berubah, terjejas. So it will change the conformation of the active site. When there is a change in the conformation of the active site, we can say that the enzyme is denatured. When the enzyme is denatured, the substrate cannot bind to the active site. And that is the end of the story. So dia bergantung kepada jenis enzyme, apa optimum pH dia. Asalkan terlalu rendah ataupun terlalu tinggi, dia tidak dapat laksanakan dia punya reaction. Okay, so that is all that I want to talk about for pH for now. We're going to come back to pH again later in this lesson. So just a very quick summary. These are the properties of enzymes that you should remember. That they are globular proteins, they are required in small amounts, they are not altered at the end of the reactions, they are specific to their reactions, they could be reversible, and they are affected by temperature and pH. So I hope it's been clear so far. We're going to move on to the next segment in this subtopic. So this next segment, we're going to be talking about the classes of enzymes. So we can divide these enzymes into several classes based on what is agreed on by the Union of Biochemistry and Molecular Biology, or as we shorten it, IUBMB. So these scientists in IUBMB, they classify enzymes based on the reaction it catalyzes. The first class is hydrolases. The second class is isomerases. Third class, transferases. Fourth class, lyases. Fifth class, oxidoreductases. And sixth class, ligases. Now, there are 6 here, and there is actually a very simple way to try and remember the acronyms, which is HIT and LOL. So when you think of HIT, you think of hydrolases, isomerases, transferases, and when you think of LOL, you think of lyases, oxidoreductases, and ligases. Now let's have a look at each of these classes of enzymes. We'll start with lyases, because this is probably the more simple one you can think of. To lyase means to add or remove chemical groups other than by hydrolysis. So ini kita biasanya untuk pecahkan sesuatu molekul lah. Boleh juga untuk tambahkan sesuatu chemical group. Tapi untuk lyases, dia tidak akan melibatkan molekul air. It's not going to involve the formation of water molecule or using a water molecule to break something. So example of lyase is adenylyl cyclase. The second class of enzyme that we have here is hydrolases. So hydrolases will catalyze the hydrolysis by using water of a substance. So this one will involve using a water molecule to break a substance. Examples of hydrolases are esterases, proteases, glycosidases, nucleosidases, and lipases. Some of these you will actually see a few more times, such as proteases and lipases. The third class of enzyme that we'll look at in this slide is oxidoreductases. And if you can guess from the name, it's going to involve oxidation and reduction reactions. So it involves the transfer of oxygen and hydrogen atoms between the substances. So these are going to include all oxidation reduction reactions. Examples of oxidoreductases are peroxidases, hydroxylases, oxygenases, and reductases, and also dehydrogenases. The fourth class that we'll look at in this slide is ligases. Ligase, as you remember from DNA ligase, their function is to join things together. So they will form covalent bond between two molecules by using energy from ATP. The fifth class is transferases. As you could probably guess from the name, transferase will transfer a functional group from one substance to another. So examples of transferases are transaminases and phosphorylases. And last but not least, we have isomerases. If you remember what the word isomer means, then you could probably guess its function. Isomerases will catalyze the rearrangement of atoms in a molecule by converting one isomer to another isomer. So the first example is isomerase, and another example is phosphoglucoisomerases. Dan biasanya nama dia ada isomerase juga lah. Okay, so that is just a very brief overview of the classification of enzymes. Fairly simple, fairly straightforward, and that can take us to the next segment. So in this next segment, we talk about how the enzymes will lower activation energy by making reactions fast, easy, and efficient. Now, if you don't get every single sentence in this segment, that's okay. I would like you to use this segment as mainly to think of how you can answer essay questions that is connected to this question. So, ini lebih kepada membantu kamu menjawab soalan lah. Sebab kamu, dalam otak kamu sudah faham yang enzim itu akan mempercepatkan reaction. Cuma slide-slide berikutnya akan membantu kamu untuk explain saja. Okay, so how do enzymes lower the activation energy? Before a reaction can occur, the reactant or substrate must reach the transition state. So the amount of energy required to reach this transition state is called activation energy. And once again, we are looking at this graph here. Activation energy is talking about this part of the curve. Enzymes lower the activation energy by forming enzyme-substrate complex. Macam mana dia dapat turunkan activation energy? Sebab dia membentuk ini, enzyme-substrate complex. When it forms the ESC, it brings substrates together into the correct orientation. So, dia akan mempercepatkan proses dengan membawa substrate sama-sama dalam susunan yang betul. It will also facilitate the breakage of old bonds. So, dia akan bantu pemecahan bond lama. Facilitate the formation of new bonds. Membantu pembentukan bond baru. And the enzyme also orientates the substrate and helps in the progress of the reaction. So, enzyme ini dia akan bantu untuk susun lah, untuk pastikan substrate berada dalam kedudukan posisi yang betul untuk mempercepatkan reaction. So, we can also see this in the form of diagrams over here. On the left, you have a substrate that is being broken into two products. So, on the left, the function of the enzyme substrate complex is that it will weaken or break the bonds, and the substrate will be changed easily. The second part, the second diagram on the right, is showing how you can take two substrates and fuse them together to form one product. In this case, the enzyme substrate complex will facilitate in making the bonds and bringing the reactants together. So we have this example down here. In this example down here, we have this enzyme that will facilitate in the formation of fatty acyl-CoA. So before the reaction starts, we have CoA, we have fatty acid A. The function of the enzyme is to form enzyme-substrate complex, brings them together, brings them close, facilitate the breaking of old bonds and the formation of new bonds, and last but not least, produce the reactants, the products. Okay, so this is how the enzymes will lower the activation energy as illustrated in diagrams. I'm not sure if you would be asked to draw any graphs anytime soon, but just in case, this is some graphs for you to see. So this is also the same point that I talked about earlier. It's just shown in a diagram form. So if you need to explain, for example, you could start by saying, first step, substrates will enter the active site. The enzyme will change its shape such that an active site will enfold the substrates in accordance with the induced fit model. And as you can see from the second step here, the substrates are held in the active site by weak interactions, such as hydrogen bonds and ionic bonds. The active site can lower activation energy and speed up a reaction by acting as a template for substrate orientation, stressing the substrates and stabilizing the transition state, providing a favorable microenvironment, and or participating directly in the catalytic reaction. So that is the third part in this note here. After that, the substrates are converted into the products are released, and the active site is now available for 2 new substrate molecules. So that is what is being shown in this slide here. Oke, itu sahaja yang kita perlu tahu untuk how enzymes lower activation energy. So, satu benda yang kamu nampak di sini yang mungkin menarik minat kamu adalah perkataan induced fit. Okay, the enzyme will change its shape in order to enfold the substrates, which brings us to this next segment where we talk about the mechanism of action of enzymes according to the induced fit model. Now, this is the induced fit model or induced fit hypothesis. The hypothesis is the active site of the enzyme will have a shape which does not exactly fit for the substrate. The enzyme and its active site is actually flexible. That means mula-mula dia punya bentuk tidak sama, tapi nanti bila itu substrate masuk, dia akan berubah bentuk dia. It's gonna change its shape in order to fit the shape of the substrate. Dia akan ubah bentuk supaya dia punya corak, bentuk, shape, ngam dengan substrate. Before this, we used to teach you about 2 different models for how enzymes work. We used to teach you about induced fit and the lock and key hypothesis. Kalau lock and key hypothesis, dia akan cakap bahwa setiap enzyme akan ada bentuk yang ngam-ngam, sama dan betul dengan substrate yang dia mau lekat. And induced fit will be what we teach you here. But after scientists did more research and they do more studies and they do more experiments, they found that the induced fit hypothesis or the induced fit model is more suitable to explain enzymatic reactions compared to the lock and key. Jadi dalam banyak kajian, banyak perbincangan, lock and key hypothesis sudah tidak diterima. Dan sekarang kami cuma ajar kamu pasal induced fit hypothesis sebab ini yang paling sesuai dan paling betul setakat yang kami tahu. Okay, so let's learn about this induced fit model. First things first, according to this hypothesis, the substrate fits into the active site of the enzyme to form the enzyme substrate complex. The binding of the substrate will induce conformational changes in the enzyme that will maximize the fit and force the complex into a more reactive state. And then the reaction occurs. So, maksud ayat pertama itu saya sudah explain. Substrate masuk ke dalam active site. Dia membentuk enzyme substrate complex. Second point, apa yang dimaksudkan di sini, binding of the substrate induces conformational change in the enzyme. Bila itu substrate masuk, dia akan trigger, dia akan induce, dia akan menyebabkan perubahan pada bentuk enzyme. Substrate masuk, menyebabkan perubahan bentuk enzyme. Dan ini akan maximize fit. Bila enzyme berubah bentuk, it will maximize fit, so dia akan berubah bentuk dia untuk kasih paling ngam, menjadikan bentuk dia paling ngam, paling sesuai dengan substrate, and force the complex into a more reactive state. So, bila dia punya bentuk sesuai dengan itu substrate, ngam dengan itu substrate, the enzyme substrate complex is going to be more reactive. So, dia lebih bersedia untuk jalankan reaction. And then your reaction will occur. Your products will be formed, and then your products will be released. After the products are released, the enzyme will return to its original shape, and it is free to bind with another substrate. So that is it. If anybody asks you to explain what is the induced fit hypothesis, you can explain to them using these 5 steps. All right, so now we're going to talk about something else that is related to the shape of the enzyme, which is the active site and the allosteric site. Every enzyme has an active site for its substrate and a competitive inhibitor to bind. So, setiap enzyme ada active site. Active site itu boleh masuk substrate, boleh juga masuk competitive inhibitor. Every enzyme also has an allosteric site for non-competitive inhibitor to bind. Selain daripada active site, setiap enzyme juga akan ada allosteric site. Allosteric site boleh dimasuki oleh non-competitive inhibitor. Kamu boleh faham allosteric site ini sebagai mana-mana site selain daripada active site. So you could have many allosteric sites, but one active site. But what we're going to focus on more is about the concept of inhibitor. Without the inhibitor, the substrate is able to bind to the enzyme's active sites and end products will be produced. Kalau tiada sebarang inhibitor, biarpun dia competitive inhibitor atau non-competitive inhibitor, substrate dapat masuk dalam active site dan menghasilkan product. Active site kadang-kadang digelar sebagai catalytic site, as you can see from this diagram on the right. If the non-competitive inhibitor is present, it will bind to the enzyme's allosteric site, thus alters the enzyme's active site so substrate can no longer bind and no end products are produced. Maksud ayat sini, kalau ada non-competitive inhibitor, which is this pink molecule over here, dia akan masuk allosteric site dan dia akan menyebabkan substrate tidak dapat masuk dan tiada reaction. Kalau ada competitive inhibitor, if there is competitive inhibitor, it will bind to the active site and prevent binding of substrate. So here we have an animation. So this is enzyme without any inhibitors. The substrate can go into the active site of the enzyme and the products can be formed. Whereas in the second diagram over here, we show an enzyme with a non-competitive inhibitor already fused. So the non-competitive inhibitor enters into the allosteric site. When you have the inhibitor in the allosteric site, the substrate of the enzyme cannot go into the active site, and therefore no reaction will occur. So kamu boleh tengok di sini, there is already conformation change in the active site. Substrate is not able to bind to the active site. Okay, so that is very quickly what is called the induced fit model and also the concept of active sites, allosteric sites, and also inhibitors. So that concludes one segment, and we're going to go into the final segment for this subtopic, which is talking about the factors that affect enzymatic reaction. Now, what factors will be bothering the reactions? So we have 4 factors that could affect the rates of reactions. The first is enzyme concentration. The second would be substrate concentration. The third will be temperature, and the fourth will be pH. Now we're going to look at each of these factors one by one. Let's have a look at the first factor with enzyme concentration. So in the situation where you keep the pH and the temperature at a constant, as enzyme concentration increases, your reaction rate will also increase until up to this point, it's because more enzymes are present and thus they provide more active sites to form enzyme substrate complex for a reaction to occur. But further increase of the enzyme concentration could have no effect on the reaction rate because all substrates are already converted into ESC. So what is shown in this graph here? Kalau kita kasih tambah enzyme concentration, rate of reaction ataupun enzyme activity akan semakin meningkat. And this is at a point when not all active sites of enzymes are occupied. Up until a certain point, as we increase enzyme concentration, it's not going to have any effect on the rate of reaction because after this point, all the active sites of enzymes are occupied. So now we have extra enzymes but no free substrate. So that's why the reaction rate does not increase. So this is it for the first factor. Now we'll have a look at the second factor, which is substrate concentration. If the pH and the temperature are kept constant, as the substrate concentration increases, reaction rate also increases because there is more formation of ESC, which is enzyme substrate complex. But when all substrates are used to form ESC, further increase in the substrate concentration have no effect on the reaction rate because all enzymes are already used to form ESC. So as you can see from this graph here, kalau kita semakin tambah substrate concentration, rate of reaction akan semakin meningkat. This is during the time when not all active sites of enzymes are occupied. But if you keep increasing the substrate concentration, after some time, the rate of reaction is not going to increase anymore because all the enzymes are being used. All the active sites are occupied. So now you have extra substrates but no free enzymes to catalyze the reaction, which is why the reaction rate is already at a maximum. So that is it for the second factor. Now we move on to the third factor, which is temperature. Now this is something we talked about on the earlier segments of this lesson when we talked about the properties of enzymes. So we know that each enzyme functions optimally at a certain temperature. If the pH, enzyme, and substrate concentration are kept constant, the reaction rate will increase with increase in temperature until the optimum is reached. This is because there is more collision between the enzymes and the substrates, as the enzymes have a higher kinetic energy and move faster when it is in higher temperature. But further increase of temperature will decrease the reaction rate and lead to the destruction of the enzyme because enzyme is a protein. So here we are showing it on the graph once again. As we increase the temperature before optimum temperature, enzyme will have higher kinetic energy and move faster. So more enzyme substrate collision and more formation of enzyme substrate complex. The rate of reaction is doubled for every rise of 10 degrees Celsius, and the optimum temperature of most mammalian enzymes will be between 37 degrees Celsius to 40 degrees Celsius. After 100 degrees Celsius, enzymes will be destroyed. And at extremely low temperature, the enzymes will be okay, but enzyme activity stops because the kinetic energy at very low temperatures is too low. Okay, so that is it for the third factor. And now we move on to the fourth factor, which is pH. Each enzyme will function optimally at a particular pH, also something we talked about in earlier segments of this lesson. If you keep the enzyme and the substrate concentration and the temperature at a constant, the pH, when you change the pH away from the optimum value, so either the pH is too low or too high, it will decrease the reaction rate or enzyme activity. pH changes will change the enzyme molecule shape, and then the protein denatures, and also cause the active sites of the enzymes to take the charged group ions, and therefore the substrate will be unable to bind to the active site. So this is shown over here, the graph effect of pH on the rate of an enzyme controlled reaction. And here is a table showing the examples of enzymes and also their optimum pH. Once again, these are enzymes that we can find in humans and most mammals. So we have pepsin at optimum pH of 2, sucrase at optimum pH of 4.5, enterokinase at optimum pH of 5.5, salivary amylase at optimum pH of 4.8, catalase at optimum pH of 7.6. So you could see, once again, on this table, the difference in optimum pH for different enzymes. And also, this graph is here to remind you of how the rate of reaction can change with change in pH. So that's it. Those very simple explanations are explaining the factors that can affect enzymatic reactions. Ito saja lah, setiap slide itu saja explanation dia. And also here, you could see the different graphs that would represent each of these different factors. Okay, so what should you do now? You should come up with your own notes for subtopic 4.1 and then complete the related questions in Tutorial Questions, Chapter 4, Part B, Questions 1 and 2. That's all for this segment. Thank you very much for your time and your attention, and I will see you again soon. Happy learning! Bye!